it is my pleasure to introduce Dr. Ana Benitez Lopez. She is Ana, are you online right now? I just need to double check everything's working good here.
Yes, I'm here.
All right, great. Then I'll do my introduction here. So I'm so excited to host. She's one of my favorite scientists because she does things that I do much better. She's based in Spain. She did her undergraduate degree in Spain at the University of Cordoba, did her master's in the Netherlands, and then her PhD at the University of Castilla-La Mancha, which I'm pretty sure is in Madrid. Is that right?
South of Madrid.
South of Madrid. And now she currently holds a Juan de la Cerva Incorporación postdoctoral grant. I'm sure I messed that up. My apologies. And she's based at the Estación Biológica de Doñana. And so she's in Spain right now. She's tuning in bright and early for her. She just woke up on Tuesday morning. It's been Tuesday for a while over here. And Ana has known for her approaches at very large scales. So she uses meta-analyses and predictive approaches to look at changes in biodiversity at a global scale using very large data sets. And so what her results really add to the literature is a synthesis of many different site-specific results. And I think that's what she'll be talking about today. And I want to have plenty of time for questions. And so with that, I will turn it over to her and say thank you in advance for being here bright and early with us.
Yeah, thank you, Matthew. Thank you for the invitation. And thank you, everybody, for joining in. So I guess Matthew already introduced me and basically what I do. So I will just jump into the presentation. I will share my screen first. Let me see. Should be this one. Right. So today I'll be talking about the work I've been doing in the past years, first in Radboud University in the Netherlands and later on in Doñana Biological Station here in southern Spain. And I'm now funded by the Ministry of Science and Innovation of the government of Spain. So I want to acknowledge that already. So the outline of the presentation is this one. First, I'm going to be talking about how to quantify and predict hunting-induced defaunation across the tropics, and also what consequences defaunation may have for vegetation composition in the tropics, which is my latest project. So for those of you that are not familiar with the term, defaunation is basically the loss of species and populations of wildlife, or the local declines in the abundance of individuals due to human activities. And one of the main signatures of this process of defaunation is that it's affecting mostly the large size species. So this is a pretty nice figure from Rodolfo Dirso and others back from 2014, where they show how the species that went extinct in the Pleistocene are those with larger body mass, those that are extinct now in the Anthropocene, the era that we are living now, according to some scientists, are also are smaller than those in the Pleistocene, but are still larger than those that are threatened now or non-threatened. And the main causes of this of defaunation, the main drivers are habitat modification, so land use change and transformation, but also direct exploitation, so hunting. So humans have been hunting for millennia, particularly in tropical forests, which is where I'll be focusing. Problem is that nowadays it's not sustainable anymore because roads are being established and built across previously remote areas. That means that there are areas that are becoming more accessible for hunters, and therefore they can hunt pristine wildlife populations. There's also been a massive increase in the growth of human populations that are demanding more and more wild meat and body parts across the tropics overseas. There's been a modernization of hunting techniques. So there are hunters that do not hunt with bow and arrow anymore, but they use shotguns, rifles, other techniques which are much more effective and efficient. That means that they can hunt more pieces per unit time. And that means that they can hunt not only for subsistence, not only to feed their families, but also to commercialize part of the meat in local markets and elsewhere. So some estimates are of about 65 million animals extracted per year, which is around 5 million tons per year in the Congo Basin. But as you can see, in other parts across the tropics, the extraction rates are pretty high, like in Indonesia, the Amazon, or Madagascar. So what we did in 2017 was to try to use human accessibility as a proxy for hunting pressure. And this is because humans are, central place foragers. That means that normally they gather resources in the vicinity of the areas where they live, or settlements, if they build roads, or from roads. And this means that the pressure is higher in the proximity of settlements. It becomes lower as we go farther into the forest. And this means that animal densities are usually lower in the proximity of hunters' access points, and they reach almost carrying capacity as we go farther away from settlements. So using this concept and taking advantage of a large amount of study that have been looking into densities or species abundance, different distances from settlements and roads in hunted and unhunted forests, we did a meta-analysis, and we estimated that approximately 58% of the bird populations and 83%, birds amount populations are reduced by 58% and 83% in hunted areas compared to unhunted areas. We also estimated how animal abundance decline in relationship with the distance to hunters' access points. And what you see on the plots down here is the effect size, the response ratio. Positive values mean that the animal abundance are higher than in unhunted areas, whereas negative values mean the opposite. And you see differences in the size of the points because they are weighted by the precision of the estimates. I'm not going to get into that, but basically what you have to take away from this is that bird populations are depleted within 7 kilometers from hunters' access points, whereas in the case of mammal populations, these distances can go up to almost 40 kilometers away. So next question that popped in my head was like, okay, so can we use this information to actually predict across the tropics hunting-induced defaunation across the pantropical forest zone? Can we see differences in the populations of a small, medium, and large-sized species? So this was a challenging task, of course. Probably some of you know that if you want to track deforestation, basically what we can do is just use remote sensing and satellite images. And with that, you can see how land use change, how these fishbone patterns form in the tropics because of the road building and extraction of wood. But in the case of hunting, in many cases, it occurs basically in areas like this, where from the sky, what you see is an undisturbed tropical forest. The problem is that because of hunting pressure, this forest may be empty or half empty, which is a term that Redford already coined in 1992. It means that mammals and birds could be quite reduced in this kind of forest because of hunting pressure. And this is something that we cannot see from the sky. We really have to go into these areas to quantify these losses. So because we cannot monitor all the pantropical forest zone. That's why I decided to use, or I tried to see by using the studies that we have so far, we could build predictive models to have a first estimate of the defaunation across the tropics. And besides that, I wanted to compare the defaunation maps that we were gonna get with two initiatives that try to or attempt to map intact forest ecosystems or intact system across the globe. And these are the maps from Pop et al., which are the ones on the left, and of course the Human Footprint Project, which you guys probably know well over there. And these two initiatives are very nice, very powerful, but they don't take into account overhunting per se. So what we did is like we used the information from the previous paper. We expanded the database up to 160 something studies assessing local hunting impacts, and that cover almost 300 mammal species. And we collected data on over 3,000 mammal abundance response ratios. These are, if I didn't explain it well before, I will repeat it again, but these are basically the ratio between the abundance of the species in a hunted area compared to an unhunted control area at the species level. So we couple this information with spatially explicit predictors. We had the distance to rural settlements, which we knew that it was an important predictor, but we included other in the models, such as human population density, which would give us a proxy of demand for wild meat. Also the travel time to major towns, which give us an idea of how close the areas are to major towns where the wild meat could be commercialized. So then the closer to these areas, the higher the, sorry, the hunting pressure will be expected. So we also included other predictors that had to do with the reliance of local communities on wild meat to feed, basically. So in this case, it was the prevalence of stunting, which is undernourishment in children, and also how accessible livestock biomass as an alternative to wild meat was for these communities. And finally, the protection status of the area. So I'm going to show now a very complicated model scheme of how we did this, where we don't have to go deep into this, and we can. But basically, what we did was to couple all this information with these socioeconomic variables. We also included species traits, such as body mass or gild. And then we model on the one side local extirpation, so the probability of a species to become extinct because of hunting pressure, and then the abundance decline on the others separately. And then we combine these models. This is called a hard model. And this some cross-validation and reprojected per species across the tropics using ICM mammal ranges, and combine the species-specific defaunation maps into composite defaunation maps due to hunting. So what I want you to understand here is that the output that you're gonna see in the next maps is called the defaunation index, and it goes from 0 to 1. And it's basically derived from back transforming this response ratio, which was declining abundance, and then subtracting the response ratio of 1. So then we have a high defaunation index indicate a fully defaunated area, whereas low values indicate intact population. Also, I would like to show you the main drivers of hunting pressure on mammals. So we have, again, the distance to hunters access points. What you see here is, again, the response ratio, with mammal abundance becoming larger in hunted areas when it's positive and lower when it's negative. What you see here is what happens for species of different body size. So for the larger mammals, what we see is that they are quite reduced or their abundances are reduced within the proximity of hunters' access points, but the smaller species become more abundant, and mostly this is because they are released from predation or competition from other species that are hunted. Because they are not normally targeted by hunters, they basically thrive in these areas. And of course, we found that at higher human population density, the impact on mammal populations was higher. So with information, I mean, there are other drivers, but I'm showing here the most important ones. We produced the first maps of hunting-induced defaunation across the tropics, across all mammal species, regardless of whether they were hunted or not. So this is a bit how it looks like. We projected the maps for, the models for almost 4,000 mammal species. And then we aggregated these maps into this hunting-induced defaunation map, which it doesn't look that bad at first glance. I mean, you see a lot of blue, which means that these are areas that are not defaunated. But what we see is that approximately 50% of the pantropical forest zone is under some degree of defaunation. Right, so this includes small species, those that are not hunted, which actually account for a large number of the species in the tropics. But what happens when we look at the medium-sized and large-sized species, which are the ones that are actually hunted? So the picture changes. It becomes much more yellowish and redder in certain areas, especially for large-sized species. And overall, what we see is that in the case of medium-sized species, they could be defaunated around 60% of the area, and this number increases to 80% in the case of large-sized species. So we're losing these larger species across the tropics in those areas that you see. We also use this information to produce maps of hotspots of defaunation, where what we did is just to select areas where species have defaunation index of 0.7. That means that their abundances were reduced over 60%, 70% at least. So what you see is that there are areas in West Africa, which are pretty red, also south of India, Malaysia, South China, and also in Central America, where they could be considered hotspots of defaunation. And these are areas where we can see, we can find these species which are threatened. And one of them cases that is more striking is, of course, the giant ground pangolin that, well, probably you know that is one of the most, pangolins in general are the most trafficked species nowadays, because the scales are being used for Chinese traditional medicine. So finally, as I mentioned before, I compared our defaunation maps with the Intact Forest Landscape Initiative. On the top plot, you see the comparison with the Human Footprint Project. So in dark green are the wilderness areas, and lighter green is the pantropical forest zone. We see that around 11% of these intact ecosystems is under some degree of defaunation across the species. And in the case of the intact forest landscapes, this number goes up to 9%. But if we compare this with larger species, the numbers are much higher. We see that approximately 60 to 50% of the intact forest ecosystems, according to these two initiatives, could be would have large mammal populations that are defaunated. So this was the work that I published last year, and something that was bugging me already for for a couple of years is okay. So we are losing these species, and these species have major roles in tropical ecosystems. So what are the consequences of this change in animal communities for tropical forests, for the ecosystem functioning of tropical forests? Around 90 or to 80% tropical plant species depend on animals for seed dispersal and for forest, for basically regeneration. So this is a nice graph from Carolina Bello and others from a couple of years back. And what they simulated was, if we lose frugivores, what implications this could have for the dispersal of seeds and for regeneration, and also for carbon storage. So I wanted to take a more empirical approach to this question. What I've been doing is gathering data on defaunated and non-defaunated areas based again on local studies, and comparing seed dispersal and seed establishment in these areas. But before, I want to quickly explain how this process occurs. So basically, you have in tropical forests, also in other forests, right? But we're focusing on tropics. So you have primary seed dispersals, which are feeding on fruits, and they disperse them away. And then you may have some animals that predate on these seeds, which have a negative impact on the regeneration capacity of the species. But some of these predators, sometimes they bury the seeds in other areas, so they could act as secondary dispersers. And also dung beetles, also they rely on mammal dung, and they could also disperse it somewhere else. Right? And then once you get to the seedling stage, you may have herbivores feeding on these plants. If an intact community, right? But if we go to a defaunated forest, all these processes alter, because maybe you lose some of these primary seed dispersals, some of these tapirs, then the seed predators may become overabundant because of ecological release from competition and predation, and herbivores may be reduced because they are being hunted. So along all the phases in the plant cycle, they could be altered in defaunated forests. So as I said, I wanted to know how fruit removal, seed density, seedling studies, all these processes change in defaunated versus intact communities, and if this would have major implications for the forest regeneration across the tropics. So as I briefly mentioned before, I've been collecting data in defaunated versus intact forests on the numbers of fruits removed, vegetation rate, secondary seed removal, seedling density, etc. And also, and again, producing these response ratios. So the case of fruit removal would be the number of fruits removed in a defaunated forest compared to a control forest. And so far, this is an ongoing project. So these are still preliminary results, but I'm really thrilled to share them. So far, I've been collecting data across the tropics. You see that in the Americas, in South America, and Central America, there are quite some more studies done in Africa or Asia. So I hope if you guys know more studies in Asia or those areas and you want to share them, it would be really nice. So these are the sample size that I have. I have 55 pairwise comparisons for fruit removal, up to over 1,000 for seedling and sampling establishment. Or sorry, sampling or sampling. So what I'm finding so far, again, the response ratio to the negative values means that there's been a reduction, positive is an increase. So what I'm finding so far is across all these stages, we are seeing a decrease in seed dispersal services in defaunated forests. So there's less removal of fruits from the trees, there's less density of seeds on the ground. Even predation is becoming lower. There's less secondary removal, and establishment is also more reduced in the faunated forest. This is only part of the story. I mean, there's a lot to tell here, but what we would expect in non-defaunated forests is that the green shades here represent the seed shadow, so that the seeds are dispersed far away from the trees, and that in half defaunated or defaunated forests, these seed shadows become more reduced and more seeds fall under the tree. And this is problematic because under trees, these are areas where seeds become attacked by pathogens and by invertebrates, and they become non-viable. And even if they germinate and they get to the seedling phase, because there's an aggregation of seedlings, there's density-dependent competition, and not all of them reach the adult stage. So I also compared how dispersal and establishment changes away and under the tree, the parent tree, in defaunated versus non-defaunated forests. So what we are seeing is that in purple, you see what's going on as distance is far away from the parent tree. And we see that there's a massive reduction on the density of seeds. There are way less seeds being dispersed away from the tree, and there's less seedlings being established. Whereas under the tree, there's been an increase. Most of the seeds have fallen below the tree. Finally, in another study that I'm collaborating with some Brazilian researchers, we're looking at how secondary removers such as dung beetles would be affected by defaunation. And what we see here is that in green, you see the abundance of dung beetles, and in orange, the richness. In the case of hunting, in very intensively hunted forests, intensely hunted and lightly hunted forests, there's a reduction in abundance of dung beetles, because basically if mammal populations are not there, if mammals are not there, there's no dung, and basically there are no beetles that can disperse the seeds. But also in areas where there have been heavily fragmented with smaller patch size. So when there are small fragments, the abundance of dung beetles is much lower than in medium fragments or large fragments. So this is where I'm working now. My next step is basically to try to understand how all these processes are affecting forest regeneration in tropics, what consequences this could have for the carbon storage capacity. This is a map of tropical forests. This is a map from Soto Navarro and colleagues from last year, where they estimated carbon stocks in the tropics, and well, not across the world. And you can see is that in the tropics is where we see the higher carbon density. So if we are losing animals that are dispersing the seeds, sometimes very large seeds, which lead to larger trees with larger storage capacity, what could defaunation could imply for carbon stocks in the future? So to recap a bit the talk, what we have seen is that the proximity to settlements and roads is a major driver of hunting-induced defaunation, because hunting pressure is higher in these areas. 50% of the pantropical forest zone is under some degree of defaunation, and that this number could go up to 80% in the case of large mammals. That forest coverage alone is not necessarily indicative of ecosystem intactness, that we can see similarly undisturbed forests that are actually half devoid or totally devoid of mammal populations, and that defaunation seems to be profoundly altering forest regeneration in tropical forests because of the loss of animals that play major roles in the dispersal and establishment of seedlings. So with that, I would like to thank you again for having me. These are the references to the main papers I discussed today. And if you want to contact me anytime, these are my email addresses and my Twitter handle and my ResearchGate profile. Thanks a lot.